Cytochrome P450 are a superfamily of enzymes containing heme as a cofactor that mostly, but not exclusively, function as monooxygenases. In mammals, these proteins oxidize steroids, fatty acids, and xenobiotics and are important for the clearance of various compounds, as well as for hormone synthesis and breakdown. They are also important for the biosynthesis of defensive compounds, fatty acids, and hormones in plants. P450 enzymes have been identified in all kingdoms of life, including animals, plants, fungi, protozoa, bacteria, and archaea, as well as viruses. In this video, we will look at the makeup of these enzymes, how they work in the human body, and with which drugs they interact. The information presented in this video should not be used for diagnosing or treating a health problem. Consult your healthcare provider before making any changes to your health plan. Remember to subscribe to this channel so you can be notified of more pharmacy and medicine-related videos. The Cytochrome P450 or CYP450 is a superfamily of membrane-bound hemoprotein isozymes with distinct classifications. While present in most body tissues, Cytochrome P450 enzymes predominantly occupy the liver, intestines, and kidneys, with the highest concentration in the liver. Of the total 57 isozymes discovered to date, 6 of these are responsible for 90% of drug metabolism. These 6 isozymes include CYP1A2, 2C9, 2C19, 2D6, 2E1, and 3A4, with 3A4 being the isoenzyme that affects most products. Various drugs work on different isozymes, and determining which isozymes are affected is critical in drug development. The term P450 is derived from the spectrophotometric peak at the wavelength of the absorption maximum of the enzyme at 450 nanometers, when it is in the reduced state and complexed with carbon monoxide. On the top right of the slide, you'll see the cytochrome P450 catalytic cycle. The reaction sequence starts at the top with the P450 resting state having a ferric 6-coordinate low-spin heme iron axially coordinated by cysteine thiolate and a weakly bound water molecule. Binding of the substrate displaces the water ligand to leave 5-coordinate high-spin heme iron. This is reduced by a redox partner, and the ferrous iron then binds dioxygen to form a ferric superoxo complex. A further single electron reduction by the redox partner generates the ferric peroxo intermediate, which is protonated to form the transient ferric hydroperoxo species. This ferric hydroperoxo species, or compound 0, is further protonated and dehydrated to form the ferryl oxoporphyrin radical cation species compound I. Compound I is considered to be the major oxidant in P450 reactions and abstracts a hydrogen from the RH to produce a substrate radical prior to rebounding the hydroxyl to the substrate to form hydroxylated product, the ROH, and to restore the resting state of the P450. The double-headed arrow crossing the cycle between the substrate-bound ferric 450 and compound 0 describes the catalytic mechanism and related peroxygenase P450s. Direct interaction with the H2O2 produces a reactive iron oxo species compound 0 that is further protonated and dehydrated to form compound I, leading to either fatty acid substrate decarboxylation, which is the major route, or fatty acid hydroxylation, which is the minor route. On the bottom right, you'll see the typical structure of CYP450, exemplified by the oxy-BTI, the CYP165B7 from Tychoplanin biosynthesis, showing the preponderance of A helices in the structure labeled AL. A-L, together with one B sheet. The core 4 helix bundle of the P450 is shown in deep red, with the exception of the I helix, that is shown in turquoise. The regions comprising the active site are shown in colors, the BC loop region in pink and F helix shown in orange, the G helix shown in gold, I helix shown in turquoise, B strand following the K helix shown in green, and the C terminal loop shown in black. The central heme moiety is shown as sticks. In humans, the CYP450 is primarily membrane-associated protein located either in the inner membrane of the mitochondria or in the endoplasmic reticulum of cells. They metabolize thousands of endogenous and exogenous chemicals, including potentially toxic compounds, including drugs and products of endogenous metabolism, such as bilirubin, principally in the liver. It's present in most tissues of the body, and it also plays an important role in hormone synthesis and breakdown, cholesterol synthesis, and vitamin D metabolism. The mechanism of CYP450 is as such. The Cytochrome P450 enzymes undergo a multitude of reactions, each with slightly different variations depending on the enzyme and substrate involved. As monooxygenases, CYP's primary role is the addition of an oxygen atom to the substrate. In general, the mechanism of CYP exhibits in catalyzing reactions depends on a few simple steps. First, CYP450 enzymes consist of an active site made of a heme iron center, as we saw in the previous slide. The iron compound is bound to the protein by a cysteine thiolate molecule. Substrates bind to the active site at the heme group, which induces a conformational change of the enzyme's active site. Reductases are then responsible for electron transfer from NADPH. Oxygen binds to the ferrous heme group following the reduction of iron. Another electron is added via reduction, creating a iron or FeO2 group known as a peroxide state. The peroxide group is short-lived as it gets protonated twice to release water and another compound known as CYP450 compound 1 or FeO3. This activity allows for the acquisition of the necessary and further hydroxylated to a more excretable hydrophilic compound. In general, the result of the mechanism appears as such, as described in this chemical equation. Drugs and other compounds that already have a free polar group can skip phase 1, where they will proceed directly to phase 2 for conjugation. Cytochrome P450s are the major enzymes involved in drug metabolism, accounting for about 75% of the total metabolism. Most drugs undergo deactivation by P450s either directly or by facilitated excretion from the body. However, many substances are bioactivated by P450s to form the active compounds like the antiplatelet drug clopidogrel and the opiate codeine. This is when prodrugs are converted to the active drug metabolite by the CYP450 enzymes. Many drugs may increase or decrease the activity of various P450 isozymes, either by inducing the biosynthesis of an isozyme, which is enzyme induction, or directly inhibiting the activity of the P450 enzyme. A classical example includes the anti-epileptic drug phenytoin, which induces CYP1A2, 2C9, 2C19, and 3A4. Effects on P450 isozyme activity are a major source of adverse drug reactions, since changes in P450 activity may affect the metabolism and clearance of various drugs. For example, if one drug inhibits the P450-mediated metabolism of another drug, the second drug may accumulate within the body to toxic levels. Hence, these drug interactions may necessitate dosage adjustments or choosing drugs that do not interact with the P450 system. Such drug interactions are especially important to consider when using drugs of vital importance to the patient, drugs with significant side effects, or drugs with a narrow therapeutic index. But any drug may be subject to an altered plasma concentration due to altered drug metabolism. Nicotine is a compound that induces CYP1A2, and both nicotine and caffeine serve as substrates to the same CYP1A2. This explains a phenomenon of a potential mechanism behind the increased tolerance of caffeine among smokers. Many substrates for CYP3A4 are drugs with narrow therapeutic index. Again, CYP3A4 is the isozyme that affects more drugs than any of the others. Drugs with narrow therapeutic index include amiodarone or carbamazepine. Because these drugs are metabolized by CYP3A4, the mean plasma levels of these drugs may increase because of enzyme inhibition or decrease because of enzyme induction, and their effects will alter very quickly because of the narrow therapeutic index. In the next 3 slides, we'll provide more information on the 6 different isozymes, which are the primary inducers and inhibitors of the Cytochrome P450 system. The first is the CYP1A2. Common inhibitors include amiodarone, cimetidine, ciprofloxacin, fluvoxamine. Inducers include carbamazepine, phenobarbital, rifampin, tobacco, specifically nicotine, and the substrates, which means the products that would be affected by the induction or the inhibition would be caffeine, clozapine, and theophylline. So how to read this is that if you have a patient that is taking amiodarone, amiodarone will inhibit the cytochrome P1A2 enzyme. By inhibiting the enzyme, it's going to decrease the activity of the enzyme, meaning decreasing the metabolism of the substrate. So if the patient is taking amiodarone and caffeine at the same time, the amiodarone will inhibit the activity of the CYP1A2, therefore leading to an increased amount of caffeine in the body because the enzyme is inhibited. There is less metabolism. On the other side, if a patient is taking carbamazepine and clozapine, the carbamazepine will induce enzyme activity, thus increasing the metabolism of clozapine, which is the substrate. Therefore, it would have less clozapine. You will have less clozapine in the body. Cytochrome CYP2C9. Common inhibitors include amiodarone, fluconazole, fluoxetine. Inducers include carbamazepine, phenobarbital, phenytoin. Substrates include carvedilol, celecoxib, lipizide, ibuprofen, amongst others. So it's the same idea. For example, if you are taking metronidazole, metronidazole will inhibit the activity of 2C9. And let's say if you're taking the product carvedilol, because metronidazole is inhibiting the activity, you will have an increased amount of carvedilol, which is a substrate. The next 2 CYP450 enzymes, isozymes, are the 2C19 and 2D6. A list of inhibitors, inducers, and substrates are listed here. So for the 2C19, fluvoxamine, isoniazid, ritonavir. Inducers include carbamazepine, phenytoin, rifampin. Substrates are omeprazole and phenobarb. For the 2D6, you have bupropion, duloxetine, amongst others. 2D6, ironically, there are no current identified inducers, but substrates are plenty, including amitriptyline, carvedilol, codeine, amongst others. And on this last slide, we have CYP2E9, E1, which has no inhibitors, but inducers include ethanol, which is alcohol, isoniazid, and tobacco, which is nicotine. Substrates include acetaminophen, theophylline, and verapamil. CYP3A4, like I mentioned before, is the isozyme that has an effect on most products. Inhibitors include this long list: amiodarone, amitriptyline, carvedilol, chloramphenicol, fluvoxamine, tramadol, verapamil, amongst others. Inducers include carbamazepine, griseofulvin, phenobarbital, phenytoin, rifampin, St. John's wort. Substrates, again, a large amount of products listed here: alprazolam, amlodipine, buspirone, amongst many others. So this is definitely one of those enzymes, isozymes that you definitely want to consider uh, in your practice. I hope you found this video educational and useful. Please remember to like and share. Thank you.